Eukaryotic cells. The eukaryotes include all the protists, plants, animals, and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm. Through the presence of membrane-bound organelles, eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeleton structure. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plants and animal cells are different as the former possess cell walls, plastids, and a large central vacuole which are absent in animal cells. On the other hand, animal cells have a centriole which are absent in almost all plant cells. Let us now look at individual cell organelles to understand their structure and functions. Cell membrane. The detailed structure of membrane was studied only after the invention of electron microscope in 1950s. Meanwhile, chemical studies on the cell membrane, especially on human red blood cell, enables the scientists to deduce the possible structure of plasma membrane. These studies showed that the cell membrane is mainly composed of a bilayer. Also, the lipids are arranged within the membrane with the polar head toward the outer side and the hydrophobic tail towards the inner part. This ensures that non-polar tail of saturated hydrocarbons is protected from the aqueous environment. In addition to phospholipids, membrane also contain cholesterol. Later, biochemical investigation clearly revealed that the cell membranes also possess in protein and carbohydrates. The ratio of proteins and lipids vary considerably in different cells types. In human beings, the membrane of the eukaryote has approximately 52% protein and 40% lipids. Depending on the ease of extraction, membrane protein can be classified into integral and peripheral. Peripheral protein lie on the surface of membrane while the integral protein are partially or totally buried in the membrane. An improved model of structure of cell membrane was proposed by Singer and Nicholson in 1972, widely accepted as fluid mosaic model. According to this, the quasi-fluid nature of lipids enables lateral movement of proteins within the overall bilayer. This ability to move within the membranes is measured as its fluidity. The fluid nature of membrane is also important from the point of view of functions like cell growth, formation of intracellular junctions, secretion, endocytosis, cell division, etc. One of the most important functions of the mycoplasma is the transport of the molecule across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called the passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is, from higher concentration to the lower. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecule cannot pass through the nonpolar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient from lower to the higher concentration. Such a transport is an energy dependent process in which ATP is utilized and called active transport. Example: sodium and potassium pump. Cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell wall not only give shape to the cell and protect the cell from mechanical damage and infection, it also help in cell-to-cell interaction and provide barrier to undesirable macromolecules. Algae have cell wall made up of cellulose, galactans, mannans, and minerals like calcium carbonate, while in other plants it is consist of cellulose, hemicellulose, pectinase, and protein. The cell wall of young plant cells, the primary wall, is capable of growth, which individually diminish as the cell mature, and the secondary wall is formed on the inner towards the membrane side of the cell. The middle lamella is a layer mainly of calcium pectate which hold or glue the different neighbor cells together. The cell wall and middle lamella may be transferred by plasmodesmata which connect the cytoplasm of neighboring cells. Endomembrane system. While each of membranous organelle is distinct in terms of its structure and function, many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system include endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. Since the function of mitochondria, chloroplast, and peroxisome not coordinated with the above component, they are not considered as the part of endomembrane system. The endoplasmic reticulum. Electron microscopic studies of eukaryotic cells reveal the presence of network of reticulum of tiny tubular structure scattered in the cytoplasm. That is called the endoplasmic reticulum. Hence, ER divides the intercellular space into two distinct components. That is a luminal inside endoplasmic reticulum and extraluminal cytoplasm compartments. The endoplasmic reticulum often show ribosomes attached to the other surface. Or the endoplasmic reticulum bear ribosome on their surface is called rough endoplasmic reticulum RER. In the absence of ribosomes, they appear smooth and are called smooth endoplasmic reticulum SER. RER is frequently observed in the cell actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of nucleus. The smooth endoplasmic reticulum is the major site for synthesis of lipid. In animal cells, lipids like steroidal hormones are synthesized in SER, smooth endoplasmic reticulum. Golgi apparatus. Camilo Golgi first observed densely stained reticular structures near the nucleus. These were later named Golgi bodies after him. They consist of many flat dish-like sacs or cisternae of 0.5 micrometer to 1 micrometer in diameter. These are stacked parallel to each other. Varied number of cisternae are present in the Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cis or the forming phase and convex trans or the maturing phase. The cis and the trans phase of the organelles are entirely different but interconnected. The Golgi apparatus principally performed the functions of packing materials to be delivered either the intracellular targets or secreted outside the cell. Material to be packed in the form of vessels, vesicles from the endoplasmic reticulum fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remain in close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its transphase. Golgi apparatus is the most important site of the formation of glycoproteins and glycolipids, lysosomes. They are membrane-bound vesicular structures formed by the process of packing in the Golgi apparatus. The isolated lysosomal vessels have been found to be very rich in almost all types of hydrolytic enzymes, hydrolase, lipase, lipase, proteinase, carbohydrates, optimally active site active at the acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. Vacuoles. The vacuoles in the membrane-bound space formed in the cytoplasm. It contains water, sap, excretory product, and other materials not useful for the cell. The vacuole is bound by a single membrane called tonoplast. In plant cells, the vacuoles can occupy up to 90% of the volume of the cell. In plant, the tonoplast facilitates the transport of a number of ions and other material against concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protists, food vacuoles are formed by engulfing the food particles. Thank you.